I'm sorry. It's a pleasure to introduce our next speaker, Jack Cook, who will be speaking on permeability studies from an innovator's perspective for BCS biowaiver support. Dr. Cook is Vice President in Clinical Pharmacology Department at Global Product Development at Pfizer. He holds an adjunct faculty position at the University of Michigan as well as Florida College of Pharmacy. He has authored or co-authored over 75 peer-reviewed publications. He has served as an industrial representative to the US FDA Pharmaceutical Science and Clinical Pharmacology Advisory Committee. He's a fellow of AAPS, and his current interests include improving therapy by optimizing drug delivery and the use of modeling and simulation to make rational decisions in drug development. Thanks, Jack.
Hello. Let me first start out by thanking the members of the workshop's organizing committee for inviting me here today. It is very much appreciated. As just noted, the topic of my talk is an innovator's perspective on permeability studies in drug development, and specifically those conducted for BCS biowaiver support. A brief agenda of what I plan to cover in my talk. First, I'd like to talk about the role of permeability in drug development, noting why it's important, and examples of when, where, and how it is assessed. Secondly, I'd like to talk about the recent ICH M9 efforts to harmonize criteria for BCS-based waivers, looking at the benefits of that effort. It's also an opportunity for me to talk about some aspects that I would wish would be included in the next time one tries to update such a harmonization effort, and then finally some conclusions. Well, permeability is assessed in drug development, not just for BCS classification, it has other uses as well. As the figure on the right denotes, the blue bars being reasons for attrition in 1991, poor bioavailability, poor pharmacokinetic parameters was the #1 reason compounds attrited, accounting for 40% of all attrition. Thus, industry reacted and tests were developed to assess compounds and their potential to be absorbed. One of the first ones were Lipinski's rule of 5, where by measuring physiochemical parameters and judging those against a set amount of criteria, one could be more assured that the compound would have favorable bioavailability. In fact, looking again at the figure on the right, one sees by year 2000 that the attrition due to poor bioavailability, poor pharmacokinetic parameters has dramatically dropped, falling to about 10%. There is further calculation besides the computational. Historically, things started out with low throughput methods. You'll see a seminal graph on the right that relates intestinal perfusion with the fraction absorbed in humans. Nowadays, such an assessment is done not with a low throughput via rat intestinal perfusion, but through high throughput screens, for example, cell line assays using CACO2, MDCK cells, or PAMPA. And these correlations between the amount absorbed in human and the permeability or flux remain rather high. So other than the computational, this slide depicts what might be considered as when permeability is assessed in drug development. Preclinically, through high throughput permeability assessments. As I noted before, it's predictive. It's not only predictive of the permeability of a compound, but coupled with other measurements, there is a a good correlation between prediction and actual exposure achieved in our early studies, and this has been a great benefit when trying to design and pick doses for first-in-human studies. And it also is done early on for development decisions and deciding what molecules to take forward or not. If it has a low chance of reaching levels necessary to elicit effect, there's little reason for bringing that compound forward. It is often assessed again in phase 2 for an individual compound, both as help in predictive measures, maybe for dissolution, and secondly for regulatory, that would be a pathway decision for, say, bioequivalence and a BCS biowaiver. The current practice, as I stated earlier, are high throughput cell-based assays. It is important to note that the CACO2 may not be the method of choice. Pfizer had done some work with the MDCK-LE line, found it comparable and in some cases superior in determining permeability versus other cell lines. Found it very robust, cost-effective, and easier to work with in comparison to CACO2. So there were some clear benefits, including less background transporter signals and less probability of interference. So the point of this slide is to show that high throughput screens are used, not necessarily CACO2, and this will come up a little bit later when I talk about wish list. Now, getting to how does one then later on in drug development, when one wants to consider a BCS waiver, measure permeability, current practice might look something like this. One could use urinary excretion data from a first-in-human study. The pro for such a approach is that it's already built into the studies. We're already collecting that type of data. The con is that very few compounds are excreted 85% unchanged in urine or greater. So it actually is applicable in only a small number of cases, even for BCS class 1 compounds. The second method would be a mass balance microtracer study. It's in ICH-M9. It has been universally acceptable across regulatory agencies for a while now. It does have a con in that the capacity, which is the need for the synthesis of radiolabeled compound for a study, is limited, and we find that we can only run about one of these studies a quarter and may actually have a need that exceeds that. Finally, single cell-based assays or cellular-based assays like the CACO2 are acceptable via ICH. The con is that it may not be what you've already done previously, and so it's not a paradigm that you would normally do. It's an added cost with added supportive studies and not part of our standard development. All right, I'd like to share with you the design of the mass balance study that can be used for permeability classification. In these types of studies, we use microtracer doses, and this will allow us to calculate absolute bioavailability. The studies are run as 2-period designs. In both periods, oral unlabeled drug is given. Also, labeled drug is given, either as an IV or oral. The paper noted at the bottom will give details of exactly how bioavailability and fraction absorbed are calculated, but suffice it to say that the absolute bioavailability can be looked at by comparing the PK parameters for cold drug, which would be given orally, and radiolabeled drug, which would be given IV in the first period, and that looking across the 2 periods in urine, one can get an idea of the fraction absorbed. I want to draw your attention to ICH-M9, the guidance for industry for BCS-based waivers. It was recently this year finalized, and kudos for all those involved. It is important. It establishes a best practice for industry to follow. It eliminates what I call regulatory uncertainty, and that is probably one of the large barriers within companies to proceed for a BCS-based waiver, is that unless you had a case such as greater than 85% excreted unchanged in the urine, which was universally accepted, there may be a regulatory agency where you wanted to file somewhere in the world that would require you to do or require you to do an in vivo study or human study, and thus you wouldn't pursue the waiver, because if you already have to do a clinical study, those are universally accepted, and it's not cost effective to establish bioequivalence by yet another method. The nice thing about the ICH in general, it avoids practice being set by the most conservative agency. And as I just said, if one regulatory agency requires a clinical BE study and others don't, the practice will be to always do the clinical BE study for the reasons just stated. So for all of you who worked on ICH-M9, thank you very much. There were some advances in there. The criteria to support permeability was lowered. It was considered highly permeable if greater than or equal to 85% of the doses administered, so that is slightly down from 90%. It also allows one to use CACO2, that being routinely accepted by all those who support ICH-M9. And as a result, more countries now accept biowaivers. And because it's nearing Christmas, and because I think I've been very good so far this year, I offer you the following wish list for consideration. This wish list comes from a IQ Consortium paper that commented on the ICH-M9 guidelines that were in development at the time, and we put forth several ideas that one could consider for harmonization. And one has to do with my emphasis before on CACO2 cells, where that is not always the method used by companies. There are other cell lines that are proving to be equally good, or in some cases better. And one might want to expand the use by establishing what criteria would need to be met for those methods to be considered acceptable. There are a number of cell lines and co-cultures and assay systems that are good candidates for assessing against such criteria. This is somewhat precedented, because if you look at Annex 1 for CACO2 cell permeability assay method considerations, there is a validation section within the M9 guidance that in essence validate the CACO2 setup in individual laboratories. The idea of setting up the validation criteria also has a benefit as allowing a way to keep up with the science without necessarily having to restart the ICH process. So as new improvements and methods become available, one can judge them against the criteria, whether they're acceptable or not, without having to go in and hard code a particular cell line or particular methodology within the ICH guidance. Another wish is that one should specifically state that a totality of evidence approach is acceptable. And the idea here is if a single method fails to demonstrate a permeability classification conclusively, you might be able to demonstrate that by adding additional methods or additional data, and such that in totality this data provides sufficient evidence that it's acceptable. It's also important to recognize that human data probably supersede in vitro and animal data. For example, if there are cases where a compound have been identified as substrate for efflux transporters, yet the fraction absorbed is more than 90% across the therapeutic range, the human data probably should trump the preclinical data. Now, the rationale for this is that many agencies already are open to a totality of evidence approach. Unfortunately, some are more conservative, and this results to defaulting into clinical BE trials. As I say, innovators typically are looking at global registration, doing one study that would be acceptable to all, and unless it's specifically stated in a guidance that individual agencies should be open to a totality of evidence approach, the regulatory uncertainty will probably keep innovators from taking advantage of this to the degree that it could be. I submit that it's always preferable to establish bioequivalence adequately without doing a BE trial, meaning if you can establish it to a sufficient likelihood, if you can reduce that risk sufficiently that things are bioinequivalent, it's better to do that without a clinical trial. As healthy volunteers receive no direct benefit from participating, there's only risk. Wish 3 may be the easiest to achieve. This is a note from the ICH website and other regulatory agencies' website about how many have stated that they have officially adopted the guidelines. There are a number of green check marks, but there still are a number of clocks indicating that they're still in the process of implementation. So hopefully this is something that can be taken care of quite quickly. I also have for consideration another application for BCS-based waivers, and this has to do with solutions. It's right now is only to immediate release solid oral administered dosage forms or suspensions. Yet in pediatric formulation development, it's not unusual to have a solution for pediatrics lag behind the oral solid adult formulation. So it's introduced in clinical trials often after the immediate release is already on the market. Well, if a solid oral formulation is a BCS class 1 or 3 and either rapid or very rapidly dissolving, one could consider when the BE study for the solution could be waived. And the rationale is that the same thing we've used to say that dissolution, as long as it's fast enough, doesn't matter for oral solid formulations, is because they behave like a solution. Here, we would be saying that those things that behave like a solution should be comparable bioequivalent to the new solution. Now, this is going to make some people uncomfortable, is because we're asking folks to consider two different dosage formulations, and sometimes that hasn't been acceptable. So brings me to the conclusions. Permeability is used for more than BCS classification. Its probably key role in drug development is an important predictor of human pharmacokinetics, and very useful both in assessing whether compounds should go forward, as well as picking doses for first in human. We typically assess prior to our first in human studies, and then again at times during clinical development, especially with the in the preclinical setting. It's not always assessed with methodology that is currently acceptable via ICH M9, but in ways deemed acceptable enough to consider investment on the molecule. So they still have to be pretty robust. We've got some opportunities to modify ICH M9 further. One would be by incorporating method qualification validation criteria in such a guidance to allow new methodologies to be assessed and used. One could also explicitly state that totality of evidence approaches will be routinely considered. And finally, that one could make the case for a waiver for a solution formulation of a BCS class 1 solid oral formulation. So with that, I thank you all for your attention, and I am happy to take any questions if there is time. Thank you very much.
Okay, we'll take questions during the subsequent panel discussion. Thanks, Jack, for that presentation with a point of view of innovator perspective. It's a pleasure to introduce Katja Kristan and Katja Berginc, who will be giving a generic perspective. Katja Kristan has been with Lek Pharmaceuticals since 2006. Katja started her career in drug discovery and moved to development areas as an IVIBC scientist, where she is now responsible for biopharmaceutics research. As a leading scientist, she has extensive and diverse experience in drug permeability, IVIBC, IVIVR, biowaivers, and development of biorelevant dissolution drug release methods for oral formulations and topical and nasal drug products. Katja Berginc has been with Lek Pharmaceuticals since 2013. We're focusing on innovative, cutting-edge, scientifically based approaches and developing biopredictive tools for poorly soluble compounds within emerging formulations and complex drug products. Her expertise entails dissolution, permeability, and the interplay between permeability and precipitation. Thanks for giving a lecture today.
Hello, our names are Katja Berginc and Katja Kristan, and we work in Lek Pharmaceuticals in Slovenia. We are grateful for this opportunity to present drug permeability studies supportive for BCS-based biowaivers.
In this talk, we will shortly summarize how Cockatoo cell line and rat intestinal tissue were validated in-house for the purpose of in vitro determination of permeability. And the presentation will follow with the practical demonstration of their use for the purpose of generic product development. Although rat intestine is no longer accepted in the guideline, data presented here will testify to its scientific merit in certain cases when attempting a biowaiver. The table on this slide summarizes changes since 2015. The main one addressing permeability is the omission of animal tissue from permitted models. The second one is the allowance of including certain fecal metabolites in the estimation of fraction absorbed. Altogether, one could say that with each revision, guideline is attempting to capture in vivo conditions in the experimental setup. But by omitting other permeability models, a possibility for scientifically based justification is diminished, as it will be shown later in the presentation. However, better defined conditions regarding determination of solubility and stability are highly appreciated. A correlation between the fraction absorbed and in vitro permeability needs to be demonstrated to claim suitability of the model. To comply with this request, we have determined apparent permeability for at least 5 compounds from each permeability class. That means low, moderate, and high permeability. We also included zero permeability marker. All compounds that we were testing were selected from the appendix. Sufficient number of replicates was made, at least 3. For both models shown on this slide, a good correlation was established as it is seen with high correlation coefficient. To trust measured permeabilities, one needs to prove that the system retained integrity throughout the experiment and that the permeability has not been overestimated. For this purpose, we have set 2 criteria, one for electrophysiological values and the second one for the permeability of feuilletazodextran and lucifer yellow, which are known to exert higher permeability only when dealing with damaged cells or tissue. Therefore, we have established in-house limits for transepithelial electrical resistance for KK2 cells during culturing, before and after experiment. For intestinal tissue, besides transepithelial electrical resistance, potential difference is measured in a similar manner. Figure on the bottom left side shows morphology of in-house KK2 cells when forming a monolayer. It also proves that the cells have undergone differentiation due to the microvilli on the apical side and tight junctions between the cells. On the right side, the figure shows how the limits for electrophysiological conditions were set. When tissue was deprived of oxygen, when temperature was intentionally increased, or as it was added, transepithelial electrical resistance and potential difference declined, while they remained above set-off value when tissue was kept on physiological conditions. Therefore, if both criteria are satisfied, measured permeabilities can be accepted. Even though it's not required, for both models, gene expression for transporters and enzymes were evaluated. For KK2 cells, 27 genes coding for secretory ABC transporters and 48 genes for absorptive transporters were evaluated. In rats, all investigated transporters are expressed as in humans, but the magnitude of expression is different in some cases. Overall, 65% of transporter genes are similarly expressed in rat and in human jejunum. Rhodamin 123 is a well-known PGP substrate, and with this compound, PGP functionality was assessed. On KK2 cells, where PGP expression is significantly higher than on rat, also significantly higher secretory permeability was obtained, and efflux ratio was well above 2. With the addition of PGP inhibitor verapamil, secretory permeability of rhodamin declined, and so did the efflux ratio. In rat, we checked the apparent permeability of rhodamin too. First, we checked how changing of luminal pH conditions affect efflux ratio, and then how efflux ratio changes with different subjects. Based on totality of data obtained in rat and in KK2 cells, efflux ratio of 2, as stated in the guideline, was accepted.
Levetiracetam is a typical example of a drug that can be used for BCS-based biowaiver. According to literature data, levetiracetam is highly soluble and highly permeable drug and belongs to class 1 of the biopharmaceutical classification system. It is not a narrow therapeutic index drug. It has almost complete absorption. Cmax is unaffected by food. Absorption is also not modulated via active transport. Our solubility experiments by using shake flask method confirmed high solubility of levetiracetam in pH range between 1.2 and 7.5. The highest single therapeutic dose is soluble in 250 milliliters of media. Also, the drug release is rapid, achieving nearly 100% in 15 minutes. In-house permeability experiments also confirmed significantly higher levetiracetam permeability across isolated rat intestine as compared with internal standard and high permeability marker metoprolol and low permeability marker atenolol. The permeability of levetiracetam did not differ significantly between the mucosal to serosal and serosal to mucosal directions, resulting in an efflux ratio of approximately 1, which indicates lack of transporter-mediated levetiracetam absorption. Common excipients for immediate drug release products do not affect the rate or extent of absorption, since many generic levetiracetam drug products with various compositions were found bioequivalent to the reference drug. In each case, the 90% confidence interval for AUC and Cmax was reported to be well within 80 and 125% limits. Intra-subject variability was established to be low. Taking all considerations together, levetiracetam is a very good candidate for the BCS-based biowaiver procedure. This biowaiver approach with presented permeability data was also used for drug registration in one of the market countries.
For the second case, we will present desloratadine, an antihistamine drug which has several advantages over other drugs from this therapeutic class. It is available as immediate release tablet and it is dosed in 5 mg per day. Although it has been confirmed to be PGP substrate in vitro, this transporter seems to be irrelevant for the in vivo absorption, because even grapefruit juice does not change the rate or the extent of absorption, and pharmacokinetics has been shown to be linear in the range from 5 mg to 20 mg. Data in the literature suggest high solubility as shown in the table. However, published values for fraction absorbed of 90% can be questioned. After the absorption, desloratadine is subjected to extensive hepatic metabolism, and only about 9% of unchanged drug can be recovered. The rest is recovered as metabolites in the urine and in feces, and it is unknown whether the part from the feces is formed before the absorption or after by biliary excretion. So without conclusive data regarding where the fecal metabolites are formed, data on intraluminal stability are considered incomplete. So following biowaiver guidelines, we first determined solubility. Using shake flask method, high solubility in all required buffers was confirmed after 24-hour incubation at 37 degrees. Test was performed in triplicate and pH check at the end was also done. The solution was checked under various agitation conditions. Apparatus 1 was selected at the end to avoid coning, as observed with apparatus 2 at 50 RPMs. As you can see, very fast dissolution is assured for tested tablets under all pH conditions. Data regarding luminal stability was also generated in simulated gastric fluid with and without pepsin and simulated intestinal fluid. In all three cases, the recoveries were well within acceptance limits. Permeability was assessed stepwise, using in vitro models from the least to the most complex ones. First, Pampa was used and the pH on donor site was changed to simulate intraluminal conditions. In all cases, the permeability was as high or higher than that of metoprolol, which has borderline fraction absorbed value of 85%. Since this is no longer a valid method for biowaiver request, the experimental work proceeded to the rat intestine. Tests gave
to drug conversion occurs. The second option was now removed from the guidelines. We will present the permeability of two compounds, a prodrug and an impurity named here as impurity 1, which is formed during API synthesis and is therefore also present in the drug formulation. The first aim of this study was to evaluate the rate of hydrolysis for a prodrug before and after permeation through rat intestine. For this purpose, a prodrug solution was put in a donor compartment on the left side of the using chamber with rat intestine, as shown on the figure below. Then, samples were withdrawn from both donor and acceptor compartments during the 3-hour experiment.
In this case, we will present desloratadine, an antihistamine drug which has several advantages over other drugs from this therapeutic class. It is available as immediate release tablet and it is dosed in 5 mg per day. Although it has been confirmed to be PGP substrate in vitro, this transporter seems to be irrelevant for the in vivo absorption, because even grapefruit juice does not change the rate or the extent of absorption, and pharmacokinetics has been shown to be linear in the range from 5 mg to 20 mg. Data in the literature suggest high solubility as shown in the table. However, published values for fraction absorbed of 90% can be questioned. After the absorption, desloratadine is subjected to extensive hepatic metabolism, and only about 9% of unchanged drug can be recovered. The rest is recovered as metabolites in the urine and in feces. And it is unknown whether the part from the feces is formed before the absorption or after by biliary excretion. So without conclusive data regarding where the fecal metabolites are formed, data on intraluminal stability are considered incomplete. So following biowaiver guidelines, we first determined solubility. Using shake flask method, high solubility in all required buffers was confirmed after 24-hour incubation at 37 degrees. Test was performed in triplicate and pH check at the end was also done. The solution was checked under various agitation conditions. Apparatus 1 was selected at the end to avoid coning, as observed with apparatus 2 at 50 RPM. As you can see, very fast dissolution is assured for tested tablets under all pH conditions. Data regarding luminal stability was also generated in simulated gastric fluid with and without pepsin and simulated intestinal fluid. In all 3 cases, the recoveries were well within acceptance limits. Permeability was assessed stepwise, using in vitro models from the least to the most complex ones. First, Pampa was used and the pH on donor side was changed to simulate intraluminal conditions. In all cases, the permeability was as high or higher than that of metoprolol, which has borderline fraction absorbed value of 85%. Since this is no longer a valid method for biowaiver request, the experimental work proceeded to the rat intestine. Initial tests gave so low desloratadine recoveries that permeability could not be determined. So an investigation was triggered to find the root cause for drug disappearance, since no metabolites could be quantified in the solutions. For this purpose, intestinal rings were incubated with desloratadine, and we saw that the loss of the drug from the solution can be attributed to the profound binding to the tissue, and only a small amount was lost due to unspecific binding to the plastic. Equipped with this knowledge, we evaluated desloratadine permeability using Kakadu cells because they have less biological material available for drug binding. This was also confirmed with recoveries of desloratadine well above 80%. With this model, we were able to demonstrate that permeability of desloratadine is as high as that of metoprolol. Also, we observed efflux ratio of 1.7, which confirms certain degree of PGP involvement in drug transport. Data in table also demonstrate that permeability was determined on monolayers that retained integrity throughout the test. With all generated data, we were able to demonstrate that this drug belongs to the BCS class 1.
The third case is showing permeability of a prodrug. According to previous LDS guidance document, two options were possible to determine prodrug or drug permeability, depending on site where the prodrug to drug conversion occurs. The second option was now removed from the guidelines. We will present the permeability of two compounds, a prodrug and an impurity named here as impurity 1, which is formed during API synthesis and is therefore also present in the drug formulation. The first aim of this study was to evaluate the rate of hydrolysis for a prodrug before and after permeation through rat intestine. For this purpose, a prodrug solution was put in a donor compartment on the left side of the using chamber with rat intestine, as shown on the figure below. Then, the samples were withdrawn from both donor and acceptor compartments during the 3-hour experiment. As you can see on graphs on this slide, prodrug is already rapidly hydrolyzed to drug at the donor site by intestinal brush border enzymes. This is in contrary to the literature published data claiming that the main site of prodrug to drug conversion is blood. The major compound detected in acceptor is drug, but the hydrolysis most probably occurs also at the acceptor site. It also remains unclear what is the ratio between both forms during permeation step. From the guidance, it is clear that prodrugs may be considered for a BCS-based biowaiver when absorbed as the prodrug only. However, in vivo, very seldom only a prodrug is absorbed. In such cases, permeability of drug and prodrug could be determined and, when scientifically justified, also used for BCS-based biowaiver. In the second part of this experiment, we also examined the permeability and hydrolysis of the impurity 1, which is present in drug formulation. Again, the hydrolysis of this compound to drug via prodrug occurs already at donor site, and all three, but mostly impurity 1 and drug, are detected in acceptor site. To confirm which enzymes are responsible for this conversion, we incubated impurity 1 with pure commercially available enzymes esterase and pancreatic lipase, as well as with a small piece of a rat intestine. Both enzymes hydrolyze the impurity 1 to drug, thus esterases and lipases that are present in gastrointestinal tract would rapidly hydrolyze impurity 1, most probably even before the absorption. This is an example how permeability experiments can help to interpret the fate of a potentially toxic impurity present in drug formulation. Therefore, more complex models, such as rat intestine, could be included in the guidelines to capture a wider view of drug absorption process. To conclude, BCS-based biowaiver approach is well accepted by the generic companies because it can facilitate approval for highly soluble BCS1 and BCS3 drugs. To our opinion, guidance could again allow complementary systems for drug permeability evaluation, such as retin testing and other appropriate cell lines, to capture more complex absorption features like metabolism. We also believe that BCS biowaiver approach could be applied to immediate release products containing prodrugs that convert to drugs in the gastrointestinal lumen or during permeation.
Thank you, Katja and Katja, for that perspective of from a generic company and the very informative examples. Let's see, it is now about 18 minutes before the top of the hour, so we'll take an 18-minute break and we'll resume at the top of the hour. That is at 10 o'clock Eastern Time. See you then.